talking about it. Yeah. So she lived in the 50s. Well, she died in the 50s of cervical cancer, and they took a biopsy of the cancer, and it ended up being that first line of cells that they were able to culture in the laboratory successfully. So before that, they were able to take cultures and grow them for a few divisions, and then they just kind of peter out and die. But this cell line would just grow and grow and grow. And you take a sample, you put it in a new dish, it spreads across the dish. Take a sample, put it in a new dish, it spreads across the dish. And so that's something that was unique, and it was the first time that they'd been able to achieve that in human cells. And why might that be helpful? Like, what's potentially useful about having human cells growing as much as you want in culture? Yeah. Yeah, so it basically gives you a human, like truly human cell line that you can test things on. So it's been like really a workhorse cell line. Like when I worked in a lab that did cell culture, we had HeLa cells. It was like one of the cells. We had HeLa cells, we had CHO cells, they're Chinese hamster ovary cells, and we had COS7 cells, which are like a monkey, I think a green monkey cell or something like that. And those were like 3 cell lines that we did kind of everything on, because they're super easy to grow. I mean, you just take a sample, put it in a new dish, and they spread like yeast. It's pretty wild. Yeah?
What makes for cells to keep growing?
Yeah, I think it's sort of a question that they're still exploring. But presumably it's mutations in some subset of genes, maybe combined with duplications of certain parts of the genome or deletions of certain part of the genome that lets her cells sort of avoid the normal checkpoints that non-cancerous cells are susceptible to. And they just published the genome of the HeLa cell like 2 years ago, something like that. I don't know if you guys, 2 or 3 years ago, and then rescinded it quickly because there was an outcry because they had just published the Lax family genome, not just the HeLa genome, because of course it's their family as well. So there's a huge outcry, and basically they, the same things they were doing to Henrietta Lacks family forever, they continue to do, sort of mindlessly. I wouldn't say exploit, but at least not take them into consideration in how they use the cell line. Cool. So fun, pretty cool video, or a radio piece, I think. So I want to, last time we were talking about mitosis. You guys remember that? And I want you guys just to take a moment, maybe 5 minutes or so, and go through this list of terms and discuss them with your neighbors. And make sure you all remember and are familiar with these terms. Just take a few minutes and